When one hears the words coral reefs, images of faraway places like Australia or Fiji come to mind. But what many people don't realize is that Florida is home to the third largest reef tract. The Florida Reef Tract spans from the Dry Tortugas to St. Lucie, Florida, spanning over 360 linear miles. This means that there are coral reefs right here in Broward County.
Because although they only cover 0.2% of the ocean floor, they provide hundreds, if not thousands of organisms, a place to live in their structure and also a safe place to reproduce and rear their offspring.
It's also important to study corals because they are an important economic resource to our area, providing a valuable asset for tourism, commercial, and recreational fishing.
In Broward, corals must fight off many stressors from having humans living so close to them. This includes nutrient runoff from fertilizers, which can cause algal blooms. These algal blooms outcompete with corals for space on the reef and cause habitat loss for many organisms dependent upon corals. It also can limit light available to the benthos, or bottom, which can prevent the algae which live inside the coral's tissue, referred to as their algal symbionts, from photosynthesizing.
Another stressor facing Broward corals is sedimentation. Fortunately, scientists at NOAA Southeastern are on the case.
Sediment induced from coastal construction such as dredging, beach renourishment projects, and port construction is composed of more fine-grained sediment, whereas the sediment mainly found on coral reefs is composed of coarse-grained sediment. This coarse-grained sediment allows light to be able to penetrate through the water column, which is needed for the algal symbionts within the coral tissues. Whereas fine-grained sediment can smother coral polyps, reducing their ability to feed. The smothering can also cause anoxic conditions where there won't be enough oxygen for the coral to survive.
Once a coarse sediment is applied on it, it quickly withdraws its tentacles and completely closes up, even after the sediment is removed. It may be quite some time before the coral is able to open up again. Yes, it can use its tentacles to push away sediment, but with lots of sediment, this can be quite difficult, especially since the coral, with its tentacles withdrawn, unable to catch food, and unable to expose its zooxanthellae to light, preventing it from photosynthesizing.
In order to understand how corals in Broward are being affected by sedimentation and ocean warming, we conducted two different experiments, one with Porites astreoides and another with Acropora cervicornis. For our experiment with Porites, we tested three different factors. The first was temperature, both ambient and ocean warming conditions. The second was sediment type, which was natural or anthropogenic sediment. And finally, we looked at the amount of sediment deposited, as well as its associated turbidity. And we had 30, 60, 90, and 120 milligrams per centimeter squared of sediment being deposited onto the juvenile corals, which correlated with NTUs, which is a measurement of the cloudiness of the water, of approximately 5, 10, 15, and 20 NTUs. Our next experiment was with Acropora cervicornis, and it had a similar setup, but it only looked at anthropogenic sediment. So we had our temperature treatments of ambient and ocean warming conditions, as well as sediment deposition with its associated turbidity. And there we had 0, 30, 60, and 120 milligrams per centimeter squared of sediment deposition, and their associated 0, 5, 10, and 20 NTUs. In order to conduct these experiments, we first had to collect larvae from the different species of corals. Porites astreoides are what is known as a brooding coral, meaning that they release larvae that are ready to settle, which means they'll find a suitable substrate, and metamorphose into a juvenile coral polyp. But for Acropora cervicornis, both spawning corals, which means that they release their gametes, or their eggs and sperm, into the water column. A single colony will release both eggs and sperm, as they are hermaphrodites. Once the corals release their spawn, we collect them off the surface of the water, recombine the eggs and sperm for fertilization at the proper ratios. After a few hours, they will begin to start dividing. And then after a few days, you have fully competent larvae, which means that they are ready to find a good substrate, settle, and then metamorphose into a single polyped coral. Now for Porites, we are able just to collect the larva right away, and there's no fertilization process that needs to be done. For the sedimentation aspects of our experiments, we first had to collect sediment, then dry and weigh it so that we could separate it into the different grain sizes, fine, medium, and coarse grain sediment, so that we could represent both anthropogenic and natural sediment on the reefs. After this, the sediment had to be applied to a tank so that we could determine how fast it was deposited, so that we could set up our experiment knowing how much sediment we had to add to keep it at the different concentrations that we desired. For Porites astreoides, we found that corals survived better in ambient than in ocean warming conditions. And this can be seen in the graph here. Across the bottom axis we have time, and across the y-axis we have the probability of survival. In this graph, the black lines represent corals that were in ambient conditions, and the red lines are corals that were in warming conditions. And you can see that the red lines are clearly all below the black lines. And this was true for coarse sediment as well as anthropogenic sediment.
Now, for the effect of sedimentation on the Porites astreoides babies, we found that it actually depended on the type of sediment used. So the natural sediment, which is made up of sand and other larger grain sizes, actually helped the corals survive because it provided them with a little bit of shelter, whereas the anthropogenic sediment, so the silt, actually smothered the corals and they did not have access to sunlight or oxygen, so they died much quicker.
For Acropora cervicornis, we found similar results in that corals survived better in ambient conditions, as seen by the solid lines in this graph, instead of ocean warming conditions, seen by the dashed lines. Different colors of the lines represent the different sedimentation levels. Since this study only looked at anthropogenic sediment, an increase in sediment decreased the coral's survival. Most importantly, though, we found that corals in the lowest sediment conditions survived equally in both warm and ambient conditions. Therefore, in the absence of local stressors such as sediment, corals can better cope with global stressors like ocean warming.
Currently, the Environmental Protection Agency, or the EPA, has set a standard of 29 NTUs for dredging and other construction activities. So when they surpass this level during those activities, they have to cease all operations until the level goes back under 29 NTUs.
There needs to be some regulations on the amount of turbidity allowed for different ecosystems. Mangroves and estuaries have a lot more sediment, and this turbidity of 29 NTUs may not be detrimental to the wildlife in those ecosystems, whereas our studies have been showing that 7 NTUs is the maximum allowable turbidity for baby corals not to incur negative impacts from human-induced sediment.
Also, if we limit dredging to the winter months, say November through February locally, the corals may be old enough to survive dredging, while also dredging activities and other construction activities aren't impacting the spawning season of corals, so it doesn't also affect the larvae.
This would avoid the compounding effects of warming and sedimentation.